Yeah. So I'd like to give a warm welcome to everyone. Maybe we just wait for a few seconds until all connections are established.
Okay.
Yeah. So once more, warm welcome to everyone joining us for this webinar and part of the European Microwaves Association's webinar series. So my name is Jan Borowski. I'm with Bochum University and partly with EuMA, and I'm very happy to host this webinar today, having here Dr. Ilona Piekarz from Krakow University. And just before we start with her surely very exciting talk, I would like to do some advertisements for those of you who are interested in doing an internship. So I'm sure we promoted this quite a lot during the European Microwave Week 2 weeks ago. And there is a possibility to apply for this award, boosting your scientific career with an internship at several very interesting host institutions throughout Europe. If you have any further interest in this, then please check our website, scan this QR code here that is available, and find out what's going on. Yeah, and after this very brief advertising block, I will switch over to today's topic. So I'm very happy to have here Dr. Ilona Piekarz with me, who will give us a talk on microwave biosensors for label-free bacteria detection. Ilona Piekarz is with University of Krakow in Poland, where she also did her PhD in 2018, and since 2023 she's an associate professor there. But she was at a lot of other institutions as well. In 2017, she was a visiting researcher at the Michigan State University. In 2021, she was a visiting postdoctoral researcher at University of Pavia, and again in 23 and 25 in Italy there. And she was also a visiting postdoctoral researcher at the Autonomous University of Barcelona in Spain. Her research interests are focused on the development of sensors and measurement systems in transmission line techniques for biomedical and agricultural applications. And in addition to that, she's very active in the scientific community. She's an active member of several MTT technical committees and also serves as an organizing and in the TPC committees for the European Microwave Week, the International Microwave Symposium, and the International Microwave and Radar Conference, MICA. Ilona, I'm very happy to have you here, and with that, I would like to hand over to you.
So thank you for your kind introduction. I try to share my screens because that will be very helpful to have a presentation with slides. Just give me a second. Okay, and now try to share this one. Okay, do you see my screen?
Yeah, perfect.
Okay, just give me a second because it would be nice also to see it here. Yeah, so look at you and see it here. So hello and welcome to my talk about microwave sensors for label-free bacterial detection. It's a really nice topic because I will show you how to use microwaves, so engineering, for some biological stuffs, some medical stuffs, so how to help humans. At the beginning, I want to introduce you to the topic, so tell more about biosensor itself, how it works, So generally, biosensors are constructed by two parts. The first part is okay. So the first part is a biosensing layer, which is constructed out of the specific biological molecules that can be antigens or something different, which can specifically bound the another biomolecules that we want to detect. One of such biomolecules are actually bacteria, about which I will talk, because we are using those biosensors for the detection of bacteria, and one of such bacteria is bacteria E. coli. And the second part of the biosensor is the transducer. That is the device which translates the reaction between the bacteria and the biosensing layer into the physical output signal. And based on this physical output signal that we can measure, we can detect if the bacteria exist. And the second thing, we can also estimate how many of the bacteria do we have. When we are talking about the electrical sensors, those circuits are mostly passive devices. It's because it's most simple to have passive device. Such circuits should be also single use only, so the cost of the production of passive devices are lower in general. And such structure is constructed as a dielectric layer on which we have metal. This metal is just the geometry of the biosensor. And we need to cover this metal, so the sensing part, with the biofunctionalization layer. So this layer which only knows specific type of, let's say, bacteria. And when those bacteria cover this biofunctionalization layer, is just treated as a thin additional dielectric layer on over the biofunctionalization layer. What is most important part is how to design very high sensitive biosensor. And to do that, we need to take into account two things. The first thing is this biofunctionalization layer itself needs to be designed in such a way to be very selective. Selective, that means that only specific type of the bacteria can be bound to this layer. Otherwise, we can detect anything. And we want to detect only specific type of the bacteria. And the second thing is the design of the sensor itself. So since the bacteria, since we want to detect as low as possible bacteria amount, and generally the entire surface of the sensor is biofunctionalized, and we don't know where the bacteria bound. And bacteria also like to bound in some spots, in some colonies, let's say. It's nice to have a sensor which features high sensitivity on even small changes of the electric properties of this layer covering the biofunctionalization layer, even not homogeneously, just some spots of changes of the dielectric properties. And that is actually the method to obtain a high sensitive biosensor. As I mentioned you before, me and my colleagues, we focus on the detection of E. coli bacteria, and therefore I want to talk maybe more about this type of the bacteria, because generally E. coli bacteria can be found almost everywhere. It can be found also in human intestine, and some species of E. coli bacteria are not harmful for humans. However, some species are harmful, very harmful, and the fast detection and also fast introduction of treatment is very important, because they can cause many of the diseases. There are conventional methods for the detection of this type of bacteria. For example, microbiological tests or plate count enumeration methods, which are quite accurate. However, they require time, because those bacteria need to grow. So even we can have the results even after several days. Therefore, there is a need to find some different solutions which allows us to decrease this time for the detection of the bacteria. On the market, you can find some other methods to detect the bacteria in faster way. And for example, PCR tests, which are also well known from the COVID detection, are one of those methods. They are faster. The results are even after several hours. But even though you need to have appropriate staff, qualified staff who prepare the test and analyze the results. And also, I think there is some problem with the webinar. Do you hear me?
We can hear you.
And do you see my screen also?
Yeah, but not at presenter mode anymore.
Yeah, because I had something with my computer. Just give me a second. I try to share it once again. Yeah.
So right now I'm still
It's
seeing.
not. Just give me a second.
Yeah.
It just stopped and everything was closed, and then I was not in a presenter mode.
Okay. Now it's back in the presenter.
Okay, so those PCR tests are much faster than the conventional methods, but even though the qualified staff is required and you need to have appropriate prepared laboratories, therefore there is a need to look something more low cost and possible to be used by people who are not specified staff to do the test much faster. Since we want to use the electrical sensors for the detection of E. coli bacteria, it's nice to have an overview how such bacteria looks like from the perspective of the electrical properties. And here you can see a free shell model. It's called because of the fact that the bacteria itself is divided into 3 parts, where the greater part is the protoplasm. And you can see here that the protoplasm properties are quite similar to water. So we can see that the relative permittivity is quite high, and also there is quite high bulk conductivity. So in general, we can expect high permittivity and also high losses when the bacteria bound to the biofunctionalization layer. The size of the bacteria is in micrometer range. So we want to have quite small sensors, quite small sizes to detect smaller amount of the bacteria. And that is the reason why we are using microwave biosensors, because among many of the electrical sensors, they allows us to realize the sensors in the range of the bacteria size. So there is a higher probability that we can detect the bacteria if the sensor is also the sensing elements in the size of the bacteria. And they are also very sensitive to even small changes of the layer covering the sensor due to the sensing of the permittivity changes. And the bacteria feature high permittivity, so the sensitivity should be also high. Therefore, quick detection should be possible. There is also a possibility to utilize label-free methods to detect those sensors, because we don't need external labeling. Only this biofunctionalization layer is needed to catch appropriate type of the bacteria. And since microwave biosensors are electrical sensors, we can easily integrate it with measurement equipment. On the market, there are many low-cost realization of the vector network analyzers, and there's also possibility to integrate it with appropriately designed 6-port diffractometers, so the entire measurement equipment can be realized for this purpose in a lower cost than, for example, for the laboratories that we need for PCR tests. Microwave biosensors can be generally divided into two groups when it comes to the frequency range of operation. The first group are broadband sensors. When we are designing broadband sensor, we are observing the changes of the parameters, the magnitude or the phase of the signal in a broad frequency range, and based on this broad frequency range changes, we can detect the bacteria. In the resonance sensors, we look only at single frequency points. We can look at the frequency variation of magnitude, phase, or both. The simplest way is to have one port device, so only to look at the reflection coefficient, but also transmission can be used, or both, to have double informations about the double information for the detection of the bacteria. Our goal was to design the sensor featuring the best sensitivity for the smallest possible amount of the bacteria. Therefore, for all of our solutions, we use the same biofunctionalization process and the same measurement procedure. That allows us to compare the results and show the advantages and disadvantages of the structure we are designing. And in our solutions, we are using a biofunctionalization process using polyclonal anti-Escherichia coli antibody. It's a chemical process. More information you can find in one of the paper of our colleague. This biofunctionalization layer is performed by Professor Sabina Górska. Here you can have a brief overview how it looks like. Generally, we don't do it on ourselves. The biofunctionalization, the biotechnology department is doing that for us. From our perspective, what is most important thing is that the sensor itself need to be designed or on a substrate which is not porous, or so generally it should be not porous, because this biosensitive layer is very thin. If we have porous material, in such a case we have some bumps, and there will be places where we will have this biofunctionalization layer and the places where we don't have that. So there will be no possibility to detect bacteria. And also it worked for inorganic substrate that was tested. So you can use silicone, you can use ceramics, you can use glass, that is possible. From the perspective of bacteria detection, the best metal which can be used is gold. Here you can see the CEM results of the sensor surface biofunctionalized, that is the biofunctionalization layer, and you can see how thin it is. We estimated it some time ago, and it varies depending on the batch from 10 to 100 nanometers. Here you can see how the bacteria bound to this biofunctionalization layer. You can see that that are the coli bacteria, that they are really in micrometer range. The procedure that we are using for the measurement of all of those biosensors looks as follows. So first, we send the sensors for the manufacturing. When we have it back, we measure the amount that we need for the measurement procedure. And we need to measure all of them because we need to get rid of the elements which had some manufacturing flaws. That is first thing. And we are also selecting those which have similar frequency characteristics, because that is better for the comparison purpose. Then the sensors, after measurement procedure, are sent for the biofunctionalization. They send us them back, and we measured all of those sensors also after biofunctionalization. And those measurements are really important because it's our base to which we compare then the results after bacteria binding procedure. After those measurements, after biofunctionalization, we need to divide the sensors into some groups. And just for testing purpose, to see how the sensors respond to various concentrations of E. coli bacteria, we need to have prepared, well-defined bacteria amount. So that is also what our biotechnology department is doing. And not only we need to have the coli bacteria, we also need to have a different type of bacteria than E. coli. We used Lactobacillus rhamnosus and bifidobacteria to check how selective the biofunctionalization layer is, because in ideal world, we shouldn't see any changes for the bacteria which is used as a negative control. All of those bacteria are suspended in PBS, that is phosphate buffered saline. So we also need to know what is the reference, so how the sensor responds to pure PBS. So that is our zero control. So we divide the sensors into some groups, put one group to for example 10 to the power of 4 colony forming unit per milliliter of the bacteria, another one to 10 to the power of 3 colony forming unit per milliliter, and so on. We can go as low as our biotechnology department can prepare for us the test tubes with the bacteria. So we also put one group to the zero control and negative control, and we need to wait some time for the incubate. So we need to wait some time till bacteria will catch by this biofunctionalization layer. So this time is called incubation time. I wrote it here 2 hours, but after some time we performed many of the tests, and 1 to 1.5 hours is actually enough. The greater amount of time we are waiting, the greater possibility that more bacteria bound to the biofunctionalization layer. So the changes will be greater, but proportionally to the amount of bacteria for which we are doing the tests. After this time, we need to take out all of those sensors, clean it in PBS, because we want to make sure that all impurities are cleaned from the sensors, and that we have only this biofunctionalization layer and bacteria bound to the biofunctionalization layer, nothing more, because otherwise we have wrong results. And after this cleaning, we need to wait some time to dry the sensors, because we after biofunctionalization we are measuring dry sensors. So after bacteria binding, we also need to measure dry sensors. The important thing here is that each sensor unit needs to be numerated, because even though they have similar frequency characteristics, in an ideal world they should have the same frequency characteristics, but always in manufacturing process there can be some variations. That is first thing. And also this biofunctionalization layer can vary from element to element. As I said during this talk, we observed that it can be between 10 to 100 nanometers. So it will also influence the measured frequency characteristics. Therefore, it's better to compare the results unit by unit. And in such a way, we are sure that the changes we are observing is because of the bacteria that were cached through the biofunctionalization layer. And the indicator of the bacteria presence are changes in the signal after bacteria binding process in comparison to those we measured after biofunctionalization. We designed broadband microwave biosensors and also resonant ones. And generally, for the resonance sensors, we can have more cheap measurement equipment. So from this perspective, it's better to have resonant frequency biosensors. But we also tried with the broadband ones, because they give us more information in a broader frequency range, which can be also used for better detection of especially low concentrations of bacteria, for which the changes of signal are really small. And here is the first sensor we designed and developed for the E. coli bacteria detection, and that was capacitive matrix sensor. At each cross of those transmission line, the capacitor was created. Here you can see the close-up look on such element. And each of this crossing is very sensitive to the bacteria presence. Such a construction allows us to put the sensitive regions across the entire sensing area. And since we don't know where the bacteria actually will bind, it's quite nice when it comes to the solution. The disadvantage here was that for each sensor we needed to have 25 measurements, because we measured transmission based on which the capacitor, the capacitance was calculated. However, some switches can be used to measure it in a faster way. The sensor was manufactured using EuMA's pH25 process on gallium arsenide. And this process allows us to do these bridges. So when it comes to the cost of the production was not so low as for the single metal circuits, but such construction allows us to detect 1,000 of colony forming unit per milliliter of bacteria, and the changes of the capacitance were quite high in comparison to other circuits that were available in literature. Then we measured the changes of capacitance first in a wide frequency range from 1 to 3 gigahertz, and then we calculated the mean value of capacitance in this through all of the frequencies, and that are the results. Not only we could detect the quite small amount of bacteria, because 1,000 is in 1 milliliter, because colony forming unit per milliliter means that we have 10,000, we have 1,000 of bacteria in 1 milliliter. But also you can see here that a monotonical response was observed. So this sensor can be also used to estimate how many of bacteria do we have. But since this technology is quite expensive and we are looking for something low cost, the second approach was to just realize the interdigital capacitor with the metal strips and the width between the offset between the strips, separation between the strips, featuring size similar to the bacteria. And this capacitive sensor was designed on silicon substrate and manufactured using standard microelectronic process. We used here similar procedure, so we calculate based on the transmission the capacitance in a wider frequency range, and then calculate a mean value across the frequencies, and that are the results. And unfortunately, in this case, we are observing smaller changes of the capacitance. That is first thing for this realization. And the second thing is that for higher bacteria amount, we are observing saturation effect. So it is possible to detect using the sensor 10 to the power of 3 bacteria amount, similarly as in the previous case. But the previous realization was better from the perspective of detection, because we observed greater capacitance changes. That is first thing. And the second thing, here we cannot estimate, especially for higher amount of the bacteria, what is the bacteria amount in real. Our third sensor, which we designed, which operated in a broad frequency range, is actually quite new. It's our newest design. And it is designed using CPW technique as a meandered transmission line. The sensor is a two-port structure, and In order to maximize the sensing area to entire sensor area ratio, we used Hilbert space filling curve to just maximize this very sensitive part. In this case, we don't calculate capacitance and we don't look at the magnitude changes. We are looking on the phase, but on a phase in a broader frequency range. So we are looking on the slope of this phase, and this phase slope changes are indicator of the bacteria presence. We manufactured the longer one and the shorter one. The longer one is operating at lower frequencies. The shorter one is operating at higher frequencies. But that was one of the first sensor which could be used for the detection of 100 colony forming unit per milliliter of the bacteria. The cost of the production are also quite low because it's a single layer of metal on silicon. So they are similar to a single capacitor. Okay, I think one more time a problem with the presentation because I have black screen.
Yeah, right now your desktop is.
I think it's just give me a second.
Yeah,
or not? I think it's because of the Zoom application, because I observed that previously when I tried to connect. Just give me a second. Okay, so once again, I need to share that.
Okay. Maybe just as a comment in the meantime, there is I just started a poll or a survey that you, the participants, are invited to answer. So please give us feedback about today and on the overall series. Yeah, then Ilona, just go ahead.
Okay, do you see the screen now?
Yeah, thank you.
Okay, hopefully that this is the last time, but we will see. And once again, I need to turn on the laser pointer. Yeah, so that was the first time we could detect smaller amount of the bacteria, and that was our last sensor, our last broadband microbial sensor that we used for the detection of the coli bacteria. And we also used some resonant microbial sensor for the bacterial detection. And the first one was the couple-line section, shorted, in which we use differential excitation to maximize the electric field in a small gap between the couple-line sections. The gap was in the micrometer range, just to be more sensitive to bacteria. And the sensor was also manufactured to lower the manufacturing cost using standard microelectronic process on silicon. Here you can see the photograph. That was a single resonance structure. And here you can see the CEM results, how the bacteria bound to the structure. You can see that it can bound everywhere almost. But if you look closer, the bacteria like to groups. So they are trying to form some colonies. This sensor allows us also to detect 1,000 of colony forming unit per milliliter. That was the smallest amount of the bacteria we detected. But similarly, as for the this interdigital capacitive sensor, here for higher amount of bacteria, we observed saturation effect. So for the estimation of bacterial amount, it was not possible to use that. So we thought to maybe design some different sensor which features more resonant which is composed of more resonators, so feature more resonances in its frequency characteristics. That was our second approach. Using this sensor, we had at least two resonant peaks and one resonant valley. The sensor was a two-port device, and the size was similar as previously. Actually, we chose for the second time this EuMA pH25 process and gallium arsenide substrate to manufacture it, but it was single layer structure, but we needed to have sorts of inductors. And this sensor features increased change of the resonant frequency. We calculated the mean value of the changes through all of those peaks and valleys. And it could be used for 1,000 colony-forming unit per milliliter of bacterial detection. But when we are looking for the changes of resonance here in gigahertz, in this case, we obtained smaller changes. And in this approach, we observed higher changes. So it's better for the detection because we have higher signals, higher changes, not higher signals, higher changes. So we thought to maybe design the sensor featuring even more resonances. And for that, we utilize LTCC structure because it allows us additionally to put only sensing element on the upper part of the substrate, and have inside of the substrate, because LTCC allows to use 3D structures for sensor realization, to cover those signal lines which should be not covered by the bacteria. And the sensor is a structure composed of 5 connected in parallel resonators. And in this case, we have actually 10 peaks and valleys together, which can be used for estimation, which position changes after bacteria binding process, and can be used for the estimation of the bacteria amount. Unfortunately, in this case, also the lowest amount of the bacteria we could detect was 10 to the power of 3. So the increase of number of the resonators will not cause decreasing of the number of bacteria we could detect. Here you can see the layout of the sensor. Here maybe you can see more how this signal lines are covered, because generally on this photo here are the signal lines, and you can see that in this case, when the circuit were manufactured, they are not seen here, and only the sensing part is just uncovered for the bacteria. And that is actually our last sensor we utilized for the bacteria detection. And from the perspective of the cost of the production, I think is the lowest one, because it was designed to be manufactured on glass substrate, and we wanted to have a design featuring loosened fabrication requirements, so to additionally lower the cost of the productions. And this sensor is implemented by a pair of weakly coupled step impedance resonators, those seen here, and they are really sensitive to even small changes of the electric layer covering the sensing elements. The sensing region is also quite high. You can see it here. That is manufactured single element. And we can increase the sensitivity of the sensor by decreasing the separation between the resonators. We use the sensor also for E. coli bacteria detection, but in this case, we looked not on the frequency variation, but instead of that, on the phase deviation at the resonant frequency. And here you can see the results for 10 to the power of 2 colony forming unit per milliliter, and here for the 10 to the power of 5 colony forming unit per milliliter. You can see also the photographs from the microscope, how it looks like for the 10 to the power of 2 and 10 to the power of 5. And that was the second sensor for which we could also detect hundreds of colony forming unit per milliliter of E. coli bacteria. The sensor was designed to operate in K-band, just to make the size of the circuit smaller. But the results we obtained here are quite promising, because it's a resonance structure, it's a one-port circuit, so we don't need to measure it in a transmission mode. And in this case, there is a great possibility to improve the results and go even lower with the number of bacteria that can be detected. Okay, so to conclude my talk, so we developed various types of microwave biosensors. We tried with the broadband approaches, we tried with the resonant approaches, and when it comes to the results, the best results we obtained for the sensors where we looked at the phase changes. We have such sensor operating in a broad frequency range, where we observe the slope changes of the phase. And we have also one of such sensor operating at, let's say, single frequency point, where we observe the phase deviation at this single frequency point. However, this one operating at resonant frequency is a one-port device, so that makes it even lower when it comes to the cost over the realization. As for now, we have possibility to detect 100 of colony forming per milliliter, but we have plans to do further tests to go even lower. Single bacteria detection would be a nice thing. However, another thing that needs to be considered during such biosensors realization is also a method how to measure such sensors, because as for now we measure all of them using Z probes, which is more laboratory stuff than for industrialization purpose. So in the future we also need to design appropriate reliable interfacing that could allow us to determine the S parameters just using our specialized measurement equipment, so without those Z probes and microscopes. And also readout electronics is it's needed to be rethinked, because the entire readout circuitry together with those sensors are needed, not only the sensor itself. Some low-cost realizations and specialized only for this particular utilization is also needed. So at the end, we want to have a standalone measurement equipment with the biosensors that can be used for single use only. And I think that's all from my side. And I want to also invite you for 2 microwave conferences that will be organized in Kraków. I will take this opportunity to do that. So one is organized in May in Kraków, that is a microwave and radar week. And the second one is organized in August, and it's General Assembly and Scientific Symposium. Thank you for my talk, and I'm waiting for your valuable questions.
Yeah, Thank you, Ilona, for this very interesting talk. So for those of you in the audience that have not yet seen it, there is a Q&A panel at the bottom of your Zoom window. I'm not sure whether there's an official chat. I think you can't use it, but there should be this Q&A panel. And yeah, please feel free to bring in your questions there. And there is already one which I can read to you, Ilona, and maybe you can then just answer, but I'll just repeat it for the whole audience once.
You can, yeah.
Thank you for this nice talk. My question is, what are the most promising frequencies in terms of the successful detection of bacteria? And in fact, I would have had a similar question from what you've experienced now and what you've designed. Which is the frequency of highest interest?
So actually, we done the measurements across a really wide frequency range. So starting from 1 gigahertz to K-band, so to over 20 gigahertz. But from the readout circuitry perspective, it would be better to have biosensor operating at lower frequencies, because the readout circuitry is much cheaper. So in my opinion, the frequency range about 5.8, 6 gigahertz would be the best choice.
Mm.
But the size of the sensor are also in this frequency range higher.
Yeah.
So
yeah. Okay.
That is the greatest disadvantage.
Yeah. I've been wondering, so on your final slide regarding the summary, you stated that, of course, single bacteria detection would be the ultimate end goal. I'm wondering, yeah, with all these COVID tests back in mind, how specific are the biomarkers that you use to bind the bacteria in your sensor? So are they so specific that you can be actually completely sure that there is only the one bacteria kind you're looking for?
With this biofunctionalization layer we are using is not possible. So okay. For sure, another biofunctionalization layer need to be used. With this one, if I remember correctly, it is possible to detect some tens of bacteria, but single, it need to be something different. But even if you could go to one order less, so to the 10 of bacteria, it would be a nice thing.
Okay. And maybe another question regarding the instrumentation. So yeah, when you are aiming for lower frequency ranges, yeah, you already said that there are low cost VNA solutions available there that are surely feasible from an economic point of view. I'm wondering, do you need to calibrate your setup to do the measurements? So do you need to do some VNA calibration before you can monitor the changes in your device, or can for example changes in resonant frequency be measured directly without larger system correction?
You know, here the calibration is not so important.
Okay.
If all of the measurements are done unit per unit, and we also and we only observe the changes of the parameters, then we don't need to know what is the influence of the cables and so on, because the entire time we are using the same measurement equipment for measuring the same sensor after biofunctionalization, after bacteria binding. So the change need to be only caused by the bacteria binding process.
Okay.
So therefore, even some low-cost vector network analyzers can be utilized.
Okay, that sounds promising. There is another question from the audience. Thanks a lot for this excellent talk. Is it possible to detect biomarkers in blood for rheumatoid arthritis detection using microwave biosensors?
If it is possible to manufacture this specialized biofunctionalization layer, yes, because so what we can sense is this permittivity changes. So the thing is, if we can design this biosensitive layer in such a way that we can catch something, then when it comes to the detection, it's very simple. So the question is if we can design such such biosensitive layer, but unfortunately I'm not a biotechnology guy, so I cannot answer 100%.
Okay. Yeah. Okay. Oh, there's a large one. There are several questions coming up. I'll just read them part by part. Any novel and more practical solutions to overcome the impact of environmental conditions such as temperature? So
that is actually a very difficult question, because all of the measurements we are doing need to be done in the same temperature. Otherwise, if we don't do that, we have very expensive temperature sensor, because the properties, so the bacteria have similar properties to water, and the parameters of water also change with temperature. So if we need to have the same temperature, should have very similar humidity, and that need to be taken into account. Unfortunately, there is no possibility to omit that.
Okay. Can you solve this problem by bringing in, for example, another resonator on the same substrate, so that's where you can be sure that there are no bacteria in this region, and by differential
measurements, it eliminates somehow the problem, but not entirely.
Okay. Yeah, there's still another question. What is the most appealing aspect of microwave-based biosensors compared to other biosensing technologies?
So it depends. So our greatest competitor are those sensors which are operating with light, so at terahertz range, because they can be quite low cost, and also the results are quite nice, but very often they require labeling, as I know. So you need to have some fluorescent tags and so on. So in such a case, the microwave biosensors are better, because we don't need this external labeling, and only this biofunctionalization layer is needed, and then the measurement is just performed using also when it comes to the measurement equipment, it can be low cost.
Okay. Maybe just another question came just into my mind.
Yeah.
Are there any limits in multiple using the same sensor multiple times? So you have to clean it surely or somehow. But are there any limits that the quality degrades by multiple usages?
So generally they are designed for single use only, because when the sensor is covered with the biosensitive layer. When the bacteria attach to this layer, then it's not so simple to clean it, because generally we need to use some alcohol, but when we use some alcohol, then we're also cleaning the biosensitive layer, so we don't have the selectivity. So it is possible to clean them entirely and send in once again for the biofunctionalization, so reusing is possible. But generally they are designed for single use only. So therefore this cost of production per unit should be as low as possible.
Okay. Yeah, and maybe I can just read another one. That's also surely interesting. Are there any ideas to apply artificial intelligence data analysis in this particular field to overcome some of these specific challenges? So yeah,
artificial intelligence, I think, can be used now everywhere. So maybe that will be useful for this to improve this measurement, to have it independent from the temperature and so on, because we are measuring. So we are doing those measurements. So measuring various concentrations, for example, of a coli bacteria, we are determining some calibration curves. So having this calibration curve, we know that if we are measuring then the sample, which we don't know what has inside, if the results are if the changes of the for example, resonant frequency is that, we should have this amount of the bacteria. So we don't measure everything. So if we are measuring, for example, at 25 at temperature 25 degrees, but we don't measure, for example, at the temperature 23, 22, and so on. So to estimate what should be the results for over atmospheric conditions, I think that should be useful. And also here, for example, we are measuring the response in pure PBS. We can also measure the results, I don't know, for example, in some food, so in milk, in juice, but we don't, for example, measure in apple juice, but in orange juice. So to estimate how it will behave for orange juice, for that, artificial intelligence would be also useful.
Okay, so yeah, to overcome all these somehow external problems and very application related problems, right?
Yeah.
Okay. Yeah. So thank you, Ilona, once more for giving us this very, very interesting talk. And everyone in the audience, thank you for being with us here today. If you have any further questions, I'm sure you can write an email to Ilona, and she will be happy to answer them.
You have 2 emails here. I also write email to my colleague because we are working on this topic together. So if you have any questions regarding this topic or you want to cooperate, we are very open.
Yeah. Okay. Yeah. And with this, I'd like to close this session today. And thank you very much, Ilona.
Thank you very much for this opportunity.
Bye-bye.
Bye-bye.